Thank you very much for the introduction. So the title of my talk is How do mutations translate into arrhythmias? And I would like to focus on one channelopathy, on the molecular mechanisms of voltage-gated sodium channel dysfunction in long QT3, on LQT3. To understand these mechanisms leading to action potential prolongation, we first have to understand the something about the function, structure and function of voltage-gated sodium channels. The sodium channel is composed of four homologous domains, 1 to 4, and these domains are coupled or linked by intracellular linkers. So the fourth segment of such a voltage-gated ion channel is positively charged. This is the voltage sensor. And at the resting membrane potential, this channel is in the closed state. So it's available for activation. It's not inactivated, but it's just closed. Here you can see such a cardiac action potential, and if it is in the closed state, there's no sodium current. Then if the membrane depolarizes, so upon a stimulus, the first three S4 segments move a little bit outward, so this causes then the transition to the open state. So the channel then opens, and this further depolarizes the cell. So there's a rapid increase of the sodium current into the cell. So there's a large influx of sodium, which then causes this upstroke of the action potential. So there's a change in membrane voltage due to this huge influx of sodium. And shortly after the channel is open, the fourth segment here moves, so a little bit later, and this fourth segment here is then coupled to this intracellular linker somehow, and this intracellular linker moves then towards the inner mouth of the channel and closes the channel. So there's a movement probably of this intracellular linker. This is the inactivation gate, and then the channel is inactivated. So these are two different states, two different non-conducting states. So the channel, if the channel goes from the open to the inactivated state, it closes rapidly, almost completely. So there's almost no sodium current during this period here. So then the channel must recover during the repolarization phase and enters again the closed state. If you slightly depolarize the cells, for instance by increasing potassium concentration in the blood, then the channel can immediately go into the inactivated state without passing this away, so without opening. So in long QT3, there's a so-called gain of function of the sodium channel, so somehow the open conformation is more stable. And this causes prolongation of this action potential. So my talk will be on the mechanisms leading to this action potential prolongation, not on the mechanisms leading to arrhythmia. So this is another point. So I'm not an expert on this field. I know or I can tell you something about this prolongation and the mechanisms. Here you can see these long QT3 mutations. A lot of those mutations are located in the intracellular region here, and as you can see, some of them are not characterized, but if they are characterized, then usually or often we find a so-called persistent current. We have heard yesterday something about that. If you apply a depolarized pulse, then you can measure an influx of sodium, and the sodium channel rapidly closes almost to zero. And if you measure then single channels, you see some openings at the beginning of the test pulse, and the channel is then closed. So you repeat again the same test pulse, and you see an opening and the channel closes. So at the beginning of the test pulse we have a sodium influx, and at the end there's actually nothing or little conduction. This was the first mutation known causing LQT3, it's delta KPQ. And again, you have actually a very similar current. If you see this whole cell current here, there's a large inward current, and the channel inactivates rapidly almost to zero. So it's almost closed. The point is almost, because only 3% of the sodium current remains, and this is called persistent current or sustained current. And if you analyze single channel traces, single channel conductance, you see at the beginning, of course, again, openings, but you also see reopenings later. So at the repolarization phase of the action potential, you have an inward current, and you have such long-lasting bursts of sodium channel activation. So now you can imagine that, of course, this current here may prolong the action potential. This is actually shown here on this slide. So what we learned from these single channel experiments is that there are bursts. This means the open state is more stable compared to the inactivated state. So some channels, a few percentage of the channels cannot enter this inactivated state, or they just reopen. So they enter this state and then again they open. And this causes this action potential prolongation. And finally, you have here this persistent current. If you have more than one channel, you see quite a big sodium conductance. It's small compared to the transient current, but large enough to prolong the action potential. Here you can see in this scheme where those mutations are located. What we learned from these studies and mutations is that actually it confirmed our previous data on structure-function relationships of the sodium channel. So we know since many years that, for instance, this is the so-called inactivation gate. So, of course, if the inactivation gate is mutated, then you may have a persistent current. So, many mutations are on this region, in this D3-D4 linker. Some mutations are also in the so-called inactivation gate receptor. If you have an inactivation gate, you must have protein parts that accept this linker, of course, where the linker attaches. And this is here, this S4, S5 helix or linker, and here this region here. What is also known is that the C-terminus stabilizes the inactivation gate occluded channel. So first this moves towards the inner mouth of the pore, and then this again closes the channel more tightly. And what is also known is that this S4 segment has something to do with activation-inactivation coupling. So this can nicely actually explain this phenomenon of the persistent current. So then if this persistent current is very, very important to cause long QT3, I thought, okay, then let's plot the QTC values against the persistent current, against the size of the persistent current. So here you have the persistent current as x-fold increase compared to wild type. So in case of wild type, this transient or persistent current is about 0.5% or less compared to the transient current. This means 1. So if it is increased, then we should see such a correlation, right? The QTC value should be prolonged. So I checked the literature, and the normal QTC value, or the average QTC value in these papers, were about 410. And here you can see the distribution of those spots. So each spot corresponds to a mutation. So this means that there are patients with a 7-fold increase of the persistent current showing with this QTC value. So what you can see here is there's actually no correlation. So this means you can't easily say that the large persistent current causes also a very strong prolongation of the QTC interval. That's not the case. You have some here, for instance, borderline QTC values, but large persistent currents. There are no persistent currents in some mutations here, for instance, but quite large or long QTC intervals. So what is the reason? This led to the idea that there must be alternative mechanisms of QTC prolongation. We found in a small family in Jena a mutation T1620K which causes long QT in those patients with a QTc of about 490, but the persistent current was very, very small. This is this delta QPQ channel with persistent current of 3%, but the QTc value is similar. So there must be some other mechanisms, and there are totally 3 alternative mechanisms causing this action potential prolongation. The first is a slower inactivation. So when the channel opens, it opens rapidly, then it normally closes rapidly. In many of the LQT3 mutant channels, this inactivation is much slower. So this probably alters this phase of the action potential and may have an effect on this, on the action potential, maybe via other channels. The reason is not known how this can indeed prolong here this phase, because it should act here, actually. But many of the channels, mutant channels, show such a phenomenon here. The second is a shift of the so-called steady state inactivation curve of the channel. As I told you, the channel is normally at the resting membrane potential in the closed state. At a slight depolarization, it can enter the inactivated state. At a stronger depolarization, it enters the open state. So if you, for instance, depolarize the cell from minus 100 millivolt to, let's say, minus 40 millivolt, then a few channels open. If you depolarize to 0, then all channels open. So this curve is the so-called steady state activation curve. This curve here is the so-called steady state inactivation curve, which means that the channels are available, all are available at minus 100 millivolt for activation. So they are all in the closed state. If I apply a test pulse from minus 100 to 0, so all the channels are here and they all open. If you, for instance, depolarize the cell to minus 60, if the resting membrane potential is minus 60, then the channels are not open. This voltage is not enough to open the channel, and the channels are almost closed. They are not available. So at minus 60 millivolt, they are in the inactivated state. And in many of the long QT 3 mutations, there's a so-called increased window current, which means that this curve is simply shifted to the right. And the window current is shown here as the overlap current between both curves here. In those mutant channels, this state is favored and this is less likely. This means this curve is shifted to the right. So if this curve is shifted, this means that, for instance, at minus 50 or minus 40 millivolt, the channels can be opened, but they are also already available for activation. So they are available for activation and they are open. And this voltage range is just the voltage range of the depolarization phase, right? So if the membrane, for the repolarization phase, when the membrane repolarizes, you just come down to this value and then the channels are available and they open. So this means during this phase they are open. A few channels are open, and this causes here this action potential prolongation. This is probably a very important mechanism in long QT3. The third mechanism is a faster recovery from the inactivated state. So the channels usually open, they are inactivated, and they must recover. And in many long QT3 mutant channels, we find such a faster transition from the inactivated state to the closed state. So it's more likely to have channels available for activation during the repolarization phase. So this will open the channel during the repolarization phase, and you have an inward current prolonging the action potential. You see, many of those long QT3 mutant channels are characterized by this feature. So now I thought, okay, now let's plot the same QTc values versus the persistent current, and let's include our knowledge on the alternative mechanisms. So light gray spot means there's one additional alternative mechanism. Gray means there are two additional alternative mechanisms, and black, there are three additional mechanisms. So if a channel is characterized by a persistent current and three alternative mechanisms, you have very long QTc values, or maybe here you have two additional mechanisms and very long QTc values, or if you have no persistent current, you have three alternative mechanisms maybe. So this means here that these additional mechanisms are additive, act additively to this persistent current. So the second point is that you can't see white spots. So all those mutations above this borderline value here with a long QTC show additional defects. So this suggests that the persistent current itself is not sufficient to prolong the action potential to cause the disease, actually. Because all the mutant channels are characterized by additional defects, and there's not a single mutation known that just shows a persistent current but no other defects. So is an increased persistent current sufficient to cause long QT3? There's one interesting mutation that was found in an African patient that causes a very large persistent current in the in vitro experiments, but no QT prolongation, or at least borderline QTC value. So there are no patients actually, the rodents were healthy. And this mutation was found in the father and in the mother, and inherited to the daughter. And there was no long QT in the parents, but the daughter had a severe phenotype and died, and it has a homozygous mutation. So in vitro experiments, they showed that if you inject into the oocyte 50-50 of the mutant RNA and the wild-type RNA, then you get a significant persistent current, but the individuals were healthy. If you inject only the mutant at a higher concentration, you get an increase of the persistent current. This shows that, of course, the persistent current is related to QTC, but it also shows that it is not sufficient. Okay, so now I come to my conclusions. Electrophysiological characterizations of the mutant channels confirmed our previous studies on sodium channel structure-function relationships. So if we analyze mutations, we also learn something about basic research on sodium channel structure-function relationships. So that's why we are very interested in those mutations. Many mutant channels are characterized by a defective open state inactivation, but it seems that a persistent current is not sufficient to cause long QT3 syndrome. And almost all QT3 mutant channels are characterized by at least two gain-of-function gating defects, not only by persistent current. And the last sentence here, I didn't show you this data. Some LQT3 mutant channels also show loss-of-function features. So, for instance, a faster inactivation or such a thing, but in that case, other gain-of-function defects dominate. So, that's why they have long QT3, although you find sometimes, for instance, a smaller current in the whole cell expression system or so. If you have a small transient current, but a large persistent current, you may induce long QT3, may have patients with long QT3. Okay, so thank you very much. Are there any questions?
Thank you. There are some questions from the audience. Let me understand. First of all, when we try to translate from cells to human, of course there is always some complexity. You have channels that have a gain of function. Majority, or you say that some of them might have also some parts of loss of function. And then you try to analyze how this gain of function prolongs the action potential of the cellular life. And then we try to compare that with the prolongation of the QT interval in the patient. Is there something that we should add here with the amount of muscle that they are affected? Because at the end, what we want to know is the patient is whether the QT interval is prolonged. But to have a prolonged QT interval, you need certain amount of cells repolarizing abnormally to be able to have an expression of the CT. So do we have something like that? Is it true what I'm saying, or is it?
Yeah, it's true. So these mutations, all of the mutations are heterozygous mutations in SCN5A, and assuming that we have 50% normal channels, 50% mutated channels, and NAD1.5 is everywhere in the heart. So we should have everywhere, also in the ventricles, then a mutated channel. But it is not known.
When we have this dispersion of repolarization, and we have targets, or we have, not targets, we have leads with a very prolonged QT and go another not so prolonged QT. Does it mean, can we translate that into the fact that some areas will have more effective channels?
We don't know that. It is unknown. You mean the distribution?
How can we have different QTs in the heart, different areas of the heart, if this is not related, like the supermodality in the SCN in the respiratory leaks, because you have predominance of IQ in the outflow tract of the argument. That's why you have this localization of the infections or everything for all of the long QT.
There are no data. So
one last question. How will class 1 antidiabetic drugs work? These three mechanisms that we are showing.
okay.
If like in my mind, again, in a very simple model, if we prolong the QT, because it have a persistent current, it will be something that flows this current and normalizes the QT. It's very simple. And now you are here entering other mechanisms that will be responsible for QT. How would
Eichmalin, for instance, shifts the steady state inactivation curve to the left, and other drugs too, also lidocaine, that is known. So that would reduce the window current, for instance. But at the same time, those drugs decelerate the recovery. That would actually, if it is, of course, it is accelerated, and if it's decelerated, but the deceleration is very pronounced. It should actually cause other disease, so this is unknown. But what is known is the effect on the persistent current, that such drugs often block the persistent current, but not the transient current. And those drugs often normalize inactivation without affecting the transient current. Not so, at least not in the way you block the persistent current. That is known.
Thank you. Just extending what both of you are saying, in the clinical situation, we are interested in myocardial recovery following left ventricular assist device usage. And we and others have published on this that following unloading, particularly in the patients who recover myocardial function to the extent that we remove the left ventricular assist devices, there is consistent what we call electrical remodeling. By that we mean shortening of the QTC. Now, should we be looking at, we theorized and speculated that that means that we will get less arrhythmia. Are we right in saying that? A. And should we be looking at sodium currents in these patients where there is consistent, and we watched both the QTC getting shorter, and we try to relate it to mechanical function. You were saying the amount of myocardium. That's what intrigued me, Professor Purgada.
So the patients carrying this one mutation I showed you, they are characterized by long QT3, and the cardiologists came to us. They say that Eichmalin test shortened QTC, so it should be the sodium channel. That's why we just sequence the sodium channel. The other point is at the same time those patients show a conduction problem. So there's right bundle branch block, and Eichmalin also normalized that. So we have no explanation for this, because a bundle branch block is a loss of function rescued by a drug that blocks the channel. So there's still some mystery in this field. But Eichmalin shortens it.
But sorry, can we answer my question? Should we be studying sodium channel function in our patients who are having so-called electrical remodeling and shortening in QTC? Should we?
It's difficult for me to imagine that you shorten QTC because of some effect on the sodium or some gain of function of the sodium channel, because it will, mechanistically speaking.
So what is the mechanism? Why do we see shortening of QTC consistently?
Well, probably I would go for the potassium channel. I would study potassium channel and see whether you're having some modification in the kinetic of the potassium rather than the sodium. But for the sodium, we will not expect any shortening of the QTC by any means, either for gain of function or loss of function. We will get a lot of conduction effect. In case of gain of function, like we see now for more prolongation, but thinking shortening the QTC because of changes in the kinetics of sodium.
It's shortened, I mean, it was very long and it's coming towards normal. It's not
So
in.
you mean that because it was long, it was some kind of gain of function already present, and then you are normalizing.
Yeah.
That would be more or less.
Eichmalin blocks potassium channels. So Eichmalin definitely blocks potassium channels.
Well, probably what you have to do is to see, to prove or demonstrate that effectively when you have to prolong the QTC is due to gain of function instead of some kinetics of the sodium, but it's not
And the relation to arrhythmias?
Oh, that's clear. I always explain it. Each time you have an imbalance in the currents, you can have a storm. That's clear. I mean, you can have something going from positive to negative. That's clear. So,
So that's
in
a price
electricity, there is always when you have an imbalance in current, you can have an arrhythmia.
imbalance means long QTC.
Right. I mean, some areas of the heart that are depolarized or not yet repolarized, and you have a gradient. Each time you have an electrical gradient, you might immediately start. Please.
In one of your first slides, you showed a schematic representation of the ion channel. And on the C terminal, if I remember correctly, there's a EF band domain, which one band is involved in stabilizing this closing. Yeah. EF domains or EF band domains are usually involved in calcium binding. Is there additional calcium involved?
Yes, probably. There's also calmodulin binding motif, and it's not clear how it works, but it's clear that during the action potential, calcium enters the cell, then calmodulin binds, and this calmodulin and calcium binding to the C terminus closes the channel more tightly. So always during this action potential, there's some safety mechanism for closure.
Is it known in more detail if this EFM motif is mechanically involved in stabilizing this closure?
There are some papers I can show you. Yeah, I don't have.
One more quick question. I was wondering, do we have any evidence yet for genetic variants in other ion channels that may be working in combination with the mutants we're seeing in the sodium channel to produce these effects? Technically, it would be tricky to study yet, but
there are a few such patients showing mutations in SCN5A and in the potassium channel. And only if you have both mutations, then you're sick. Yeah. But I don't know which of the 500 or so, but I remember a paper. It's a very rare case, but yes.
Because polymorphisms will likely have some effect, although they're not mutations, but they could be adding to the final phenotype.
Polymorphisms or pH value, for instance, phosphorylation status of the cell is very important to increase the persistent current. If you analyze those, everything is analyzed in oocytes or in X cells, so there's a fixed phosphorylation status. So what can we say, right? So
Thank you.
it's very different from the cardiac cells.